In this lecture, we're continuing our discussion of mitosis with some more practical application stuff, and we're going to talk about some of the mechanisms by which the cells can actually control the cell cycle, and I kind of wanted to ask you a question. What is cancer? Where does cancer come from? How do you go from a healthy cell to a cancerous cell? And those answers are sort of all wrapped up inside of mitosis. And despite the fact that you can actually see mitosis happening, you can actually use a microscope like the ones you guys have at home, which hopefully some of us will do in lab this week and actually see the process. There's a ton about mitosis that we still don't actually understand yet. And so I think, and I think a lot of researchers think, that by unlocking some of the secrets of mitosis that we'll be able to figure out how is it that cancer works, how can we treat cancer. So there are three main checkpoints that we discuss when we're talking about mitotic checkpoints. We have the G1 checkpoint. They call it the restriction checkpoint. We're just going to call it the G1 checkpoint. We have the G2 checkpoint, and we have the M checkpoint. Okay, and so in my opinion, the easiest way to remember what's happening in each of these is to think about what's going on during the cell cycle or what's about to happen during the cell cycle. So G1 and G2 obviously take place during interphase, right? Because interphase consists of G1, S, and G2. So here we have our G1 checkpoint. And what happens right after G1? Immediately following G1 is S, or synthesis phase, right? What are we doing in synthesis phase? We're making a copy of our DNA so that we can split. Sorry, this is not supposed to be just an overall review of mitosis, but right after G1, we are making copies of our DNA. So in G1, you're checking the chromosome to make sure that it is good. If you're a human being and you're trying to copy a cell, you don't want a chromosome, you don't want a set of genetics, or you don't want a genome that's messed up, that's filled with mutations and problems, or maybe there's been some sort of issue with the chromosome itself. So in the G1 checkpoint, you're checking not only to make sure that the DNA is good, because again, you don't want two messed up copies of a cell. You're also checking things to make sure that, like, yes, the cell has enough energy to undergo this duplication. Yes, we have enough of the materials required to undergo this duplication. And if G1, if your DNA is bad or if your cell is not ready, the process will be stopped. It will not enter into S phase. And there's a couple of different options that we're going to talk about later for what the cell can do. Next up here we have the G2 checkpoint. So what happens right after G2? After G2, you are entering into mitosis. You're leaving interphase and you're going into that mitotic phase, right? And so what G2 is checking is it's ensuring that that duplication that we underwent in S phase, that thing we just did, we're making sure that those chromosomes are duplicated appropriate. The next phase that we have here is the next checkpoint that we have here is the M checkpoint. The M checkpoint happens at the end of metaphase. And what the M checkpoint is checking is to make sure that your chromosomes are connected appropriately to those, we call them centrosomes. Okay? Because imagine what would happen if they weren't attached to those centrosomes, right? Because what's ultimately going to happen is those chromosomes are going to be pulled apart, those sister chromatids are going to be pulled apart into either side of the cell, right? And if we didn't have those connected to those edges and they weren't pulled apart, you would wind up with a cell with the wrong number of chromosomes. Okay, and obviously we don't want that. So the next thing that we need to talk about after we've gone through those phases is these three types of genes. We have oncogenes, genes that give you cancer; proto-oncogenes, genes that can easily turn into oncogenes; and tumor suppressor genes. And the kind of question I'm going to be asking you about these is something like this. Let's imagine you have a gene that controls the cell cycle. And this particular gene makes a protein, because hopefully you remember that DNA all codes for protein. But this particular gene makes a protein that causes the cell cycle to stop when the cell has an error on one of its chromosomes. Which of these three would you call this? I would call this a proto-oncogene because you mutate it and suddenly you no longer have this protein that is able to stop the cell cycle. So that gene gets a mutation, which leads to a non-functional protein. And this protein now allows the cell cycle to continue regardless of the number of errors. This has gone from a proto-oncogene, or a gene that could cause you to have cancer, to an oncogene, or a gene that does cause you to have cancer. Because, punchline here, cancer is just an out-of-control cell cycle. I'll probably put that in the text above this video so that nobody misses it. But cancer is a cell with an out-of-control cell cycle. That's why you wind up having things like tumors, where you just have a whole bunch of cells all growing out of control. Okay? So, proto-onco, onco. And the last category of gene is this thing called a tumor suppressor gene. So here is our tumor suppressor gene, P53. And by the way, tumor suppressor genes are kind of exactly what they sound like. They stop cells from continuing through the cell cycle. Actually, this is exactly like the example I just gave you. In this case, this is a proto-oncogene, P53. But normally P53 helps basically control cell damage. If there's too much DNA damage in the cell, P53 is going to stop those cells from continuing by sending them through apoptosis. Alternatively, it can help them to repair their DNA. Point being, P53 is going to say, okay, you cannot continue until you've figured out your DNA cell. But if P53 becomes mutated, it doesn't make a difference how much DNA damage there is. The cell cycle can just continue on and on and on and on, which leads you to a whole bunch of cells that are damaged and probably just going to continue bullying through their cell cycle as well. So this is a tumor suppressor gene, P53. This also happens to be an example of an oncogene, this mutated P53. So the punchline here is oncogenes lead you to have cancer. Proto-oncogenes are genes that, if mutated, can lead you to having cancer. And tumor suppressor genes are genes that basically help control the cell cycle. I don't want you to believe that all tumor suppressor genes, or I guess I should say, I don't want you to believe that the only proto-oncogenes are the same as tumor suppressor genes. You can also have things that encourage the cell cycle if mutated. Well, they will just kind of push the cell cycle through regardless of, oh, hey, you're not done copying your DNA, just keep going through the cell cycle. Or, oh, hey, you have a damaged cell membrane and there's a bunch of weird ions coming in. Doesn't matter, just keep going through the cell cycle. So yeah, proto-onco, onco, tumor suppressor. At the end here, I kind of wanted to mention binary fission in prokaryotes. This diagram is a little bit grainy, but it's figure 6.9 in your textbook. And basically what happens in prokaryotes, it's a little bit simpler. In essence, they copy their DNA. They make 2 copies of their DNA. The DNA migrates to either side of the cell, and the cell begins to pinch off because you have these proteins that start migrating inward, forming a cleavage furrow similar to what you see in animals. Not as interested in binary fusion as I am mitosis, although you should certainly know what binary fission is. But I probably wouldn't ever ask you about like specific proteins or this thing called the septum. What you should know is that binary fission is a similar, although simpler process than mitosis, and you wind up having 2 identical prokaryotic cells at the end.